Okay, hello everybody, here I am. This is my first ever science research video. So this is a video about some of the research I've been doing, and I hope you like it. We're going to see how it goes. Hopefully it spurs some conversation. We can talk with each other in the comments, and we can learn from each other. That's my goals for today. So if you like what I'm doing here, give me a like and subscribe on the channel, alright? Well, let's get going. My name is Mediphysman, and these are my medical physics lectures. So, today's topic: DNA damage induced by proton radiotherapy beams. How exciting is that? That does sound exciting. A lot of positive vibes, a lot of positive buzz beaming through the room here. See, those are my science jokes. So anyway, this is something I've been doing proton radiotherapy research for a long time. It's a topic that's near and dear to my heart, and this was some exciting research we've been doing over the last year or so. As far as disclosures, I have none. Nobody's paying me to do this. Nobody's sponsoring my videos. Nobody gave me any funding to do this. This was just some ideas we had, we were talking about, we didn't know the answer to, so we thought, hey, let's do some experiments, let's see what happens, right? So, my collaborators: Dr. John Derek Demery. He is a senior research scientist at the Army Research Lab. There is Dr. Noel Gardalla, retired. He is an adjunct research professor at the George Washington University in Washington, D.C. And there is Naratan Lamanchain, Naru. Naru is a good friend of mine. I've known him for a long time now. We do a lot of research together. We have a lot of great, really nerdy research talks together. He is the guy, I don't care where you are. If you're in the room with him, he is by far the coolest scientist in the room. I guarantee you that. So, what did we do for this research? Here's sort of a roadmap. Here's the journey we're going to take together. Let's talk about DNA damage. I'm going to go over everything I know about DNA damage. And don't worry, it's not a lot, so this is going to be a short section. Then we'll talk about our experiments. We thought, well, what is it we want to know? Okay, let's design some experiments. What can we do? What can we measure? And then on part 3, probably by the time we get there, I'm going to need a bathroom break. Not going to lie to you. But we get this far on our journey, we're going to talk about the results of our experiments, what we got, what was the data, and then let's have a discussion. What conclusions can we make? What did we learn? Or maybe more importantly, what new questions did this spur in our minds for our next set of experiments? So, backgrounds. What is it I know about DNA damage? This is it. This is the sum total of everything I've learned over the entirety of my career. There are direct interactions. So when we irradiate, the radiation can directly hit the DNA molecule, cause damage to that DNA molecule. There are quasi-direct interactions. So DNA has this hydration layer of tightly bound water molecules that are bound around it. The radiation can interact with one of those water molecules, knock out an electron. The electron hole left in that molecule can actually migrate from molecule to molecule onto the DNA and cause damage the structure of the DNA. And then finally, there are indirect interactions. So the radiation again interacts with the water around the DNA, breaks it apart, creates hydroxide, other radicals that can diffuse through the cellular environment to the DNA, interact with the DNA, causing damage on it. Then, these different types of damage, they cause single-strand breaks. These are sublethal breaks. They are proportional to dose. And they can also cause double-strand breaks. So 2 breaks within the DNA strand. These are lethal. These are the ones we're typically after when we are irradiating cancer to try to kill it, and they are proportional to the dose squared. Now, I know there are survival curves. We have linear quadratic models, alpha-beta ratios, LET, relative biological effectiveness. We have all these things. I've learned all about them, read thousands of papers, maybe not thousands, hundreds of papers, studied textbooks, done lots of experiments, and researched myself into these areas. But the thing about all of these is they're all dependent on this: single strand breaks and double strand breaks, and that's it. Okay, now I'm back. Sorry, I had to stop and take care of that forehead thing with the lighting that was going on, so I adjusted the lighting in the studio, did some adjusting on the camera the best I could, so now at least there's no reflection on my forehead. Of course, now it looks like I'm recording in the dark, but that's better than a shiny forehead. So we talked about DNA, what kind of damage there is, what I know about it, right? It's single strand breaks, double strand breaks. That's what happens. But then that begs the question to me, what the heck is a strand break, you know? And if you do a lot of reading, read a lot of papers like I like to do, you come across the same model of DNA damage from radiation in every paper. It's basically 2 lines or 2 cylinders or the helix structure, and they're broken. The backbone, the 2 lines are broken. Everything else seems to be intact. That's it. That's what DNA damage is. So a strand break to everybody is basically breaking the backbone of the DNA, right? Oh, this is my favorite one. Purple explosions. Yeah, that's what a double strand break is. The radiation passes through, a bunch of purple explosions happen, and voila, the backbone's broken. Double strand break. That's actually my leading theory for actually what's going on inside of there, if I had to take a guess. So the whole point of what we did with this study was what the heck happens to DNA when you irradiate it. And in this case, we irradiated our samples with proton beams. And so that's basically it. That's what I know. That's the questions when I did the research. I said, what the heck happens to DNA? What does a strand break mean? So that's what we did for this study, is trying to learn more about that type of thing. So the next step here is let's talk about the experiments we did. What can we do? What can we learn from these experiments? How do we do them? That sort of thing. So next step on our journey here, we went out, bought ourselves a bottle full of DNA, as anybody would do when you want to start a project with DNA, right? So we got freeze-dried lyophilized calf thymus DNA. I hear it comes from a Red Angus calf, from what I hear. A 2010 vintage, very good year for DNA. And we took that bottle and we produced our samples from it. So we produced dry samples and wet samples. So our dry sample, we took the water DNA mixture, I put it on a silicon wafer, a cleaned, sterilized silicon wafer, put it into a dryer in nitrogen atmosphere, and dried it overnight. So the water evaporated and left behind our film of DNA. So we have this dry film of dried DNA. And then our wet samples were the water DNA mixture that I put onto the wafer right before we did the irradiation. So the DNA was still fully hydrated and within the water when it was irradiated. Next, we took our wafers, as you can see here, and we packed them inside of this water equivalent plastic phantom for our irradiations. And we have 4 samples at 4 separate depths within the phantom. As you can see here, we start on the surface, 0 cm depth, 10 centimeters, 15.42 and 15.57 centimeters depth. Now that's the water equivalent thickness of this phantom, and it includes the thickness of the silicon wafers as well. So you can see proton 1 and 2, our last 2 samples there, one at the Bragg peak and one at about the distal 90-ish, distal 80, distal 90 range in there, as you can see from our Bragg peak here. And for our irradiations, our parameters, we used a 150 MeV proton beam, monoenergetic, a 10 by 10 cm treatment field, and we delivered 1 times 10 to the 11th protons, uniform fluence across that whole 10 by 10 field. So for the irradiations, that was then, as I said, our single monoenergetic beam. We used Ray Station 9 for our treatment plan and developed a nice uniform treatment plan for this treatment field. And so that calculated the dose for us and gave us the plan we delivered to the samples. The good thing about this Ray Station 9 system we have is we're also able to calculate the LET of the proton beam as a function of depth. So we were able to compare our DNA damage that we measured to the proton LET as a function of depth in the phantom. So irradiations, again, at our 4 different depths. And as we can see, as we go deeper in depth, one thing to remember is we start at the surface with 1 times 10 to the 11th protons, but as you go deeper, you lose protons. So we lose a little bit, and by the time we get to our last sample at the distal 80-90%, we've only got about 46-47% of the protons we actually started with. So the fluence that sample is being irradiated with is only about half of what the fluence at the surface is being irradiated with. So the other thing to remember is as you go deeper in depth, right, we start with our energy at about 150 MeV and a nice tight Gaussian energy distribution. As you go deeper and deeper, the median energy of the beam is decreasing. Also, the Gaussian spectrum of that beam is flattening out. It's getting much broader. So our energy spectrum is getting much broader. So you can see we start at 150 MeV. By the time we get down to our sample at the distal 90, our mean energy is about 16 MeV, and we have a very, very broad proton spectrum there. So if we sum all this up in a nice neat little table, we start with 1 times 10 to the 11th protons. At our deepest depth, where only 43% of those protons fluence is actually hitting the sample, our mean energy is dropping to about 16-17 MeV. And the LET, depending on whether you want just plain LET or you want LET, dose-weighted LET, which is what we calculate with Ray Station, that is going up significantly. And as you can see, the dose-weighted LET actually goes up by a factor of almost 20 as you go from the surface to the depth of the distal 90 in our sample. So let's look again at our DNA. We have our DNA sample. This is what DNA looks like. We have our sugar phosphate backbone, and we have our nitrogen base pairs. So in our molecular representation, what we see here is we have deoxyribose sugar, and we have a phosphate group, all connected together, and those repeat in the chain to make up the backbone. That's what our backbone's made of. And then we have our nitrogen bases. These are carbon, hydrogen, nitrogen bases, and you have one base on one backbone, one base on the other. They are connected through hydrogen bonds, and that becomes our nitrogen base pairs. So what we did is we measured the DNA using X-ray photoelectron spectroscopy. So we did DNA analysis with this spectroscopy on unirradiated samples. This is our control. And then we did analysis on samples at different depths that had been irradiated after irradiation. So what happens with XPS here, X-ray goes in, it kicks out a photoelectron, the X-ray is absorbed, photoelectron comes out. We put our electron detector down there and we capture and measure the energy of that electron that's kicked out. So we know the energy of the X-ray that went in because we dialed that up on our system. We know the electron energy that was kicked out. So the difference between those two is the binding energy, how much energy it took to bind that electron to that atom that's within that molecular bond. So if we do a measurement and we start counting the electrons we get of a specific energy, we see this peak, for instance. We'll get peaks, and we see here is a peak at 532 electron volts, And if you go through the literature and all the stuff that's been done over the years, we know that that is a photoelectron that was kicked off from a oxygen atom. So we know we just measured an oxygen atom within our DNA, right? And this particular peak, 532, actually comes from oxygen atoms that are bound up in specific molecular bonds. These complex bonds, carbon, oxygen, carbon, carbon, oxygen, phosphorus, carbon, hydroxyl, all of these will kick out an electron from an oxygen with that energy. So we know if we measured that, we've measured this electric bond, this type of molecular bond. So what we're doing with this measurement is we're measuring the number of molecular bonds that are present within our DNA sample. Okay, so let's look at our sample. We did XPS, electron or X-ray goes in, electron comes out. We get our peak. So that is our oxygen within the complex oxygen bonds here. We then irradiate our sample. Bam, bam, bam. Bazinga. XPS, we do it again, right? And now we see our 532 peak. What has happened? Ooh, it's gone down. It's decreased. So what does that mean? We're measuring fewer of these complex oxygen bonds after irradiation, right? Aha. So what happens? We lost oxygen bonds. These particular oxygen bonds occur in the DNA backbone. We know that we only see these types of bonds in the backbone. So what we've done is we've lost oxygen bonds in the backbone, and therefore we interpret that to say we have damaged that backbone. So in our analysis, we do our XPS measurements. We can measure carbon-carbon bonds, these single carbon bonds. They give us a specific energy profile, these peaks. Those occur in both the backbone and the base pairs. Then we have peaks that are specific to the base pair only. So carbon double bonds. These only occur in the base pairs. And nitrogen. So we can measure photoelectrons from nitrogen, and we know those only occur in the base pair. So if we see these peaks in our measurement, and we see these peaks change, we know we have done some damage, we've affected the base pairs. And similarly, we have also our backbone peaks. So these peaks, the oxygen peak and the phosphorus peak, those only occur from molecular bonds that are within the backbone of the DNA. So basically, we're measuring backbone damage, what's typically called a strand break, and we're also measuring base pair damage that's occurring in the DNA. So what did we do? How did we quantify this? We took our measured value. So we do the XPS of our sample. We look at all the different the peaks from all the different bonds, and we look at the XPS signal. We integrate across that particular peak, and that becomes our XPS value for that bond type. And then we looked at the percentage that value makes of the total XPS signal we measured across all peaks, and said, okay, our XPS bond makes up this percent of all the XPS signal that we measured for our DNA, right? So basically what we're saying is if we wanted to know what's the base pair percentage of our signal, we would take our base pair peaks, integrate those, get the number, sum them together, and then we would divide that by the integral of all the peaks summed together. And that would be our percentage of base pair signal. So we have our percentage of signals, and you can see here, so we have our percentage of both simple carbon bonds, the base pair bonds, and the backbone bonds, blue, red, green. And we measured and got those values for our control, our unirradiated samples, and then for the sample at each depth within our phantom as we irradiated it. Red plus green plus blue equals 100%, so they all sum up to 100. So here we have our control sample, unirradiated, and here you can see our proton irradiated DNA samples, and you can see how they change. There's some changes going on as a function of depth here. Okay, and when we see these changes, since we know that the three of those all add up to 100%, if one goes down, one goes up. If we see a decrease in one type, so here let's say we see a decrease in the backbone percentage, right? We see a decrease in the green, and we see an increase in the red. what we say is we've done greater damage to the backbone because it's decreased, its relative percentage is decreased relative to the others. So we've done more damage to the backbone than we have the other parts of the DNA. Okay, so those are our experiments. That's how we took the data, did some analysis on it. So let's see what happened, right? Aha, so here's our dry DNA sample. And you see the signal, or our change in bond percentages here. So this represents how much did the percentage change compared to the control. So you can see for the green, for the dry backbone damage, our backbone signal was 2% less on the surface than the unirradiated sample. And it stays about 2% less, so we're doing a lot of damage there until you get to the Bragg peak, and then suddenly, as you get beyond the Bragg peak, bam, big drop in the signal. If you look at the base pair, you see the opposite. So our backbone's going down rapidly where the base pair is going up. Basically what we interpret that to mean is we're doing more backbone damage there than we are base pair damage in our dry samples. So if you're looking at dry DNA and you irradiate it, you're going to do mostly backbone damage, we think. Aha. Sum up what I just said. I need to keep up better with my notes. So we see this constant 2% decrease in the backbone, a slight increase in the base pair percentage. Now I'm not plotting that blue line on this just to keep it from getting too cluttered, but the dry base pair plus the blue would have added up to the minus 2% you saw. So we see we're doing with dry DNA, if you remember all the way back, these are the direct effects where the irradiation has to directly hit the DNA to cause damage, basically. So when we're talking about direct effects on dry DNA, it's mostly backbone damage. However, in our wet DNA sample, we see very different behavior. So now the damage is about even, so the percentage change doesn't change much at the surface between the two, but then you see the backbone decreasing to the Bragg peak, and then beyond the Bragg peak, bam, shoots up, which is completely opposite to the base pair. So we're doing a lot of backbone damage, and then when we get beyond the Bragg peak, bam, the base pair suddenly goes down, which means we're doing a lot of base pair damage when we get beyond the Bragg peak, right into the Bragg peak region and beyond. So in this case, these could be direct effects. The irradiation directly hits the DNA. Could be quasi-direct, right? So it ionizes, kicks out an electron from a close attached water molecule, and the whole migrates over to the DNA, causing damage. Or it could be indirect effects, where we're creating radicals within the water that diffuse over and attack and damage the DNA. So all kinds of damage is at play here. So when we add these quasi and these indirect effects into the possibility, as we get into the Bragg peak and beyond, the type of damage, our damage profile, changes rather dramatically here. So now we wanted to look at this as a function of LET, as I mentioned. So this is the dose-weighted LET value as calculated by Ray Station 9 here. And this is their equation for how they're doing it within each voxel. And as you can see, again, right where the LET starts to take this sharp increase in the Bragg peak and beyond, what happens? Our DNA damage profiles increase. When it's dry, our backbone damage, our direct effects causing the backbone damage are increasing. And in the wet DNA, where we have quasi-direct and indirect also included, now our base pair damage seems to spike right when we get into this sharp increase in the LET. So now, results. What do we get from our results, right? If we look at some of the papers in the literature have discussed this, there's people who've done damage as far as double-strand breaks as a function of depth, and this is one that was published just recently, last year. There are a wealth of other papers that talk about these sorts of things, so whatever your favorite paper is, I'm sure I'm not mentioning it here, but I've read like 20 or 30 papers talking about this sort of stuff. And they all kind of have a central theme where the double-strand breaks seem to peak right at the Bragg peak and then fall off rapidly. What they did in this paper, which I liked, was they not only looked at these double-strand breaks through the gamma H2S foci induction in their irradiated cells. But they looked 24 hours after irradiation, they looked again, and what they saw was a lot of these double-strand breaks were repaired. As you can see, the numbers on the y-axis here drop significantly. So there's not a lot of double-strand breaks left. They've been repaired. But the ones that are left have shifted a little bit deeper. So they're at the Bragg peak and then beyond the Bragg peak in that high LET region. So in that high LET region, you're inducing these lethal, unrepairable double-strand breaks. And these are the ones I think we're very interested in because we want the cell to not repair them and the cancer to die. So it's these complex DNA lesions that we should be mostly interested in. And as you can see, we have here where the gray dashed line is where our Bragg peak is. And once you get beyond the Bragg peak, that's when we see this sharp increase in base pair damage, right? So our lethal double-strand breaks, as they talked about in this other paper, are correlating to the same point in space just beyond the Bragg peak as our increased base pair damage, right? So maybe that's the reason why the cell can't repair the damage is all this base pair damage, which we really haven't been talking a lot about in the literature up until now. So what we did next. So we thought this was interesting, and we say, okay, it's going up, LET's going up, our damage is changing, but we really only have like 1 or 2 points. Can we get some more points at higher LET values and see if this trend continues? So we wanted to extend our LET values. Well, the first step we did actually was extend it below, so we went to lower LET values. So what we did was actually took our DNA and irradiated it with a 6 MV X-ray beam, right? We put it at D max, about 1.5 cm depth in our phantom, and irradiated it with 6 MVs, 10 by 10 cm field, same uniform fluence, depth of D max. And so for 6 MV X-rays, the LET from the literature I found in a few papers give you an LET value of about 0.18 to about 0.2 keV per micron. So this is a lower LET value than we had with any of our protons. So here is our proton data I just showed you, but it's plotted with LET, not dose-weighted LET, just LET on the x-axis, and our percent change again on the y-axis. So it's a little different, same data plotted a little differently. But you can see we have our surface, our entrance, we have our Bragg peak, right? And then we did our X-rays, which are at a lower LET. So they were down here. So we add a couple of points, and this becomes our dry and wet X-ray sample. So you can see, especially in the wet DNA sample on the bottom, that's our wet DNA sample, the green is a little bit lower and it goes up and it reaches a maximum and it's up around the peak and then suddenly falls off. And it's kind of the opposite behavior for the base pair damage. So if we look then and we take our dry DNA sample, right, and we have our X-ray value there at 0.18, and now we wanted to extend it to higher values. So we took these to the Army Research Lab and used their Pelletron accelerator that could get lower proton values and even alpha particle beams. And we used those to irradiate our samples, and we got and compare the damage we saw. We could only do dry samples because the samples had to be under vacuum to get these low energy beams through and everything, so I couldn't do a wet sample under vacuum. So we only have data for the dry samples here. But if we have greater than 100 keV per micron LET, so we're going to add more values here. We irradiated with 3 MeV protons, 1 MeV protons, 4.5 and 3 MeV alphas, and you can see, so we extend our LET values from 12 all the way up to about 160 keV per micron. So again, here's our Bragg peak, and then we've added these new points, and you can see there's a sharp drop, but then it seems to recover as you go higher and higher and higher, back to about where it was on the surface or where it was in the Bragg peak region. And this is interesting because it's opposite of what we were seeing in the Bragg peak region, but I think we're reaching that saturation value that everyone talks about. When the LET gets too high, then you've just done too much damage in that local area, enough to cause the problem, but more damage on top of that isn't going to kill it more, right? So we're sort of losing that, and we're doing damage to both the base pairs and the DNA, and they're being almost completely destroyed. So the percent change is coming back to about a 0 value, and everything's coming back into equilibrium just because of so much damage. So these are the points for each of our extra beams we irradiated with, and so really that's what we saw. We saw this sharp change in our damage profile as you got into the Bragg peak and beyond the Bragg peak, right? And so with our dry DNA, that were direct effects. So it's direct effects only on the DNA. And I'm looking at this now, and I've just noticed I have my plots here swapped. So the bottom plot should be on the top. That's actually the dry DNA. And the top plot should be on the bottom. That's actually the wet DNA. So please forgive my mislabeling here. So anyway, if you look at the top where it's the wet DNA, not the dry like it says, that's the direct, the quasi-direct, the indirect effects are all there. That's physics and chemistry affecting what's going on to our DNA, and it causes a much different damage profile as the LET increases. If you look at the bottom plot, dry DNA, not wet like it says, so our dry DNA with only direct effects, our damage is substantially mostly backbone damage. So what we saw is as the base pair damage increases, as your LET gets roughly above 5 keV, we see an increase in the base pair damage. Yes, thank you, PowerPoint. This, what I'm talking about now, is an untested hypothesis. This is just my thoughts from what we saw in the data here. The base pair damage is increasing as we get into that region, and that's where we see increased cell death. An increased, as we call, biological effectiveness for our protons, right? So the base pair is where all the genetic information is encoded. So if we're destroying that with a much higher efficiency, then that is possibly why our proton RBE is going up, because we're doing a much more efficient job when we add in these quasi-direct and indirect effects of DNA damage into destroying the genetic information within the DNA so that it cannot repair itself and the cell cannot reproduce. So anyway, yes, I will stop talking now. I think that's about the end of everything we did. So if you have any questions about all of this work, go back, watch it again, pick out all the mistakes I made. I put at least 3 or 4 of them in there for you. Easter eggs, right? So leave me a comment. Let's have a conversation about what's going on here. Maybe you have some great ideas about the data that I didn't notice. I would love to learn and hear what you have to say. So shoot me a comment. Let me know what you think, and I'll reply to you, and then we'll see what we can learn from it. All right? Oh, and as always, as I'm legally obligated to tell you, I didn't put it on here, but please like this video and subscribe to my channel. We get a group of people going, we get a better conversation going, we all learn more, right? I could post more videos, we can have great talks, and we can learn a lot from each other. So the more you like, the more you subscribe, the more we learn, the more we put onto the channel. So anyway, I hope this was informative, and I will see you on our next video.